Hello, and welcome to Sleep Archive. To the human eye, a honeybee is a simple creature, a fuzzy, golden insect drifting lazily from flower to flower in a summer garden, a singular, insignificant speck of life. But this perception is an illusion of scale. If we shrink down, if we look past the individual and see the system, we find something that rivals the complexity of human civilization. We find a society that has mastered architecture, thermodynamics, agriculture, and symbolic language. We find a biological machine that operates with a cold, industrial efficiency, driven not by individual choice, but by a collective, chemical intelligence. A honeybee colony is not a family. It is a single, sprawling animal. It is a superorganism. It weighs 5 kilograms. It has 50,000 mouths. It has 100,000 eyes. It has a single reproductive organ, the queen, hidden deep within a fortress of wax. It regulates its own body temperature. It defends itself with a standing army. It makes life or death decisions through a democratic process of vibration and dance. In this series, we will dismantle this machine. We will pull apart the layers of the hive to reveal the biological gears turning inside. We will look at the genetics that force a sister to sacrifice her life for her kin. We will look at the physics of their flight, the chemistry of their communication, and the geometry of their construction. We are about to enter a world where the individual does not matter. We are about to enter the hive. We begin our analysis of the honeybee, Apis mellifera, not in the hive, but in the deep history of the Cretaceous period, approximately 120 million years ago. To understand the biology of the bee, we must first understand its taxonomic origin. Bees belong to the order Hymenoptera, a massive group of insects that includes sawflies, wasps, and ants. Within this order, they fall into the superfamily Apoidea and the clade Anthophila. Phylogenetically, bees are effectively vegetarian wasps. They evolved directly from a lineage of carnivorous sphecoid wasps, family Crabronidae. These ancestral wasps were predators that hunted other insects, such as thrips or aphids, to feed their larvae. The critical evolutionary divergence occurred when these wasps began hunting prey that visited primitive flowers. The prey insects were often coated in pollen, a protein-rich, granular substance produced by plants for reproduction. Initially, the wasps consumed the pollen as a byproduct of eating the prey. Over millions of years, a selective pressure emerged, favoring wasps that could digest and utilize this plant-based protein more efficiently than animal protein. This shift from carnivory hunting prey to herbivory collecting pollen marked the birth of the first bees. It was a metabolic transition that fundamentally altered their morphology and behavior. The most distinct morphological difference between a wasp and a bee is the structure of their setae body hairs. Wasps possess simple, unbranched hairs, which are aerodynamic and smooth, suitable for a predator that needs to shed dirt and debris. Bees, however, evolved plumosetae hairs that are branched and feathery at the microscopic level. These branched hairs serve a specific biophysical function. They maximize surface area to trap pollen grains via electrostatic forces. This adaptation turned the bee into a flying particulate collector. Alongside this, bees developed specialized structures for transport, such as the scopa, dense brushes of hair on the abdomen or legs, and later, in the Apidae family, the corbicular pollen basket on the hind tibiae. This evolutionary leap coincided with the Cretaceous terrestrial revolution, a period marked by the explosive radiation of angiosperms, flowering plants. Prior to this, the world was dominated by gymnosperms, conifers, cycads, and ferns, which relied primarily on wind for pollination. Wind pollination is energy inefficient. It requires the plant to produce massive amounts of pollen in the hope that random air currents will carry a grain to a female cone. The angiosperms developed a more precise delivery system: entomophily, insect pollination. By producing nectar, a sugar-rich solution derived from phloem sap plants enticed insects to visit them. In exchange for this caloric reward, the insect would inadvertently transport pollen from the male anther to the female stigma. This relationship is a prime example of coevolution. The plants and the bees drove each other's evolution. Flowers evolved bilateral symmetry, specific colors, often in the UV spectrum, and complex landing platforms to accommodate bee anatomy. Bees evolved longer glossae tongues to reach deep nectaries, specific sensory distinctiveness to identify flowers, and the cognitive capacity to remember resource locations. This mutualism is the biological foundation of the modern terrestrial ecosystem. It allowed angiosperms to become the dominant plant life on Earth, and bees to diversify into the 20,000 species we see today. The genus Apis, to which the honeybee belongs, appeared much later, roughly 35 million years ago during the Oligocene epoch. Unlike solitary bees, Apis developed advanced eusocial behaviors and the ability to thermoregulate their nests, allowing them to survive in cooler climates and persist through winters. This resilience enabled Apis mellifera to spread from its evolutionary cradle in Africa and Europe to almost every habitable corner of the globe. The fundamental unit of biology is the organism. In most species, the organism is a discrete, autonomous individual capable of survival and reproduction. However, in the case of Apis mellifera, the individual bee is not the biological unit. A single bee cannot survive outside the context of the colony for more than a few hours. It cannot reproduce. It cannot maintain homeostasis. The true biological unit is the colony itself. This organizational structure is defined as eusociality. Eusociality is the highest level of social organization in the animal kingdom. To be classified as eusocial, a species must exhibit 3 strict biological criteria: cooperative brood care, individuals caring for offspring that are not their own, overlapping generations, parents and offspring living together in the same nest, and, most critically, a reproductive division of labor. This division separates the population into 2 distinct castes: the reproductive caste, the queen and the drones, and the non-reproductive caste, the workers. This stratification transforms the colony into a superorganism. In this model, the bees are not analogous to citizens in a city. They are analogous to cells in a body. The queen functions as the reproductive organ, the ovaries. The workers function as the somatic cells, muscle and tissue, maintaining the structure and defense of the body. And the foragers function as the circulatory system, transporting nutrients from the environment to the core. The mechanism that maintains this rigid hierarchy is chemical, not behavioral. The cohesion of the superorganism relies on the suppression of worker reproduction. Workers are genetically female and possess ovaries, but under normal conditions, these ovaries remain rudimentary and inactive. This suppression is achieved through a potent chemical signal known as the queen mandibular pheromone, QMP. QMP is a complex chemical blend secreted from the queen's mandibular glands. It is classified as a primer pheromone, meaning it alters the long-term physiology and endocrine system of the recipient. rather than just triggering an immediate behavioral response, which would be a release a pheromone. The primary active components of QMP are 9-oxo-2-desonoic acid, 9-odor, and 9-hydroxy-2-desonoic acid, 9-HDA, along with aromatic compounds like methyl p-hydroxybenzoate, HOB, and homovanillyl alcohol, HVA. The transmission of this signal is physical and tactile. The queen is constantly attended by a retinue of 10 to 12 workers who groom, lick, and touch her with their antennae. As they do this, they pick up the QMP compounds on their bodies. These attendant bees then move through the hive and engage in trophallaxis, mouth-to-mouth food exchange, and antennal contact with other workers. This creates a chemical cascade. The QMP is distributed from the queen to the retinue, and then from the retinue to the general population, permeating the entire hive within minutes. Physiologically, QMP acts as an endocrine disruptor in the worker bee. When detected by the worker's sensory neurons, it inhibits the synthesis of juvenile hormone, JH, in the corpora allata, a gland in the brain. High levels of juvenile hormone are typically associated with ovarian development and foraging behavior. By suppressing JH, the queen effectively chemically castrates her daughters. Furthermore, QMP suppresses the production of dopamine in the worker brain, a neurotransmitter linked to ovarian activation. This chemical dominance ensures that the queen is the sole reproductive source for the colony. It prevents reproductive conflict, where workers might attempt to lay their own eggs. However, this control is fragile. If the queen dies, becomes ill, or simply runs out of sperm after 3 to 5 years, her production of QMP drops. When the concentration of QMP in the hive falls below a critical threshold, the chemical inhibition is lifted. The workers' corpora allata begin to produce juvenile hormone. Their ovaries swell and activate. Within weeks, workers may begin to lay eggs. However, because workers cannot mate, they can only lay unfertilized eggs. Due to the genetic mechanism of haplodiploidy, which we will cover next, these unfertilized eggs develop only into males' drones. A hive with laying workers is a hive in terminal decline. It has become a somatic body trying to reproduce without a germline. It will produce a final generation of males and then perish. Therefore, the stability of the superorganism is a constant chemical negotiation. The queen broadcasts her presence and vitality through 9-ODA, and the workers, perceiving this signal, remain in a state of physiological arrested development, dedicating their energy to the survival of the whole rather than their own genetic legacy. The driving force behind the complex social structure of the honeybee is a unique genetic architecture known as haplodiploidy. In the vast majority of the animal kingdom, including mammals and most other insects, sex is determined by specific chromosomes. In the order Hymenoptera, however, sex is determined not by the presence of X or Y chromosomes, but by the number of chromosome sets an individual possesses. In a standard diploid organism, both males and females inherit 2 full sets of chromosomes, one from the mother and one from the father. In honeybees, Apis mellifera, this rule is broken. Only the females, the queens and the workers, are diploid, possessing 32 chromosomes. The males, or drones, are haploid, possessing only 16. Drones develop from unfertilized eggs. They have no father and inherit their entire genetic identity solely from their mother. A drone is, effectively a mobile flying gamete of the queen. The molecular mechanism that controls this determination is the complementary sex determiner CSD gene. The CSD locus is highly polymorphic, meaning it exists in the population with dozens of different alleles or variations. When a queen lays a fertilized egg, the resulting zygote receives 2 copies of the CSD gene, one from the egg and one from the sperm. If these 2 alleles are different heterozygous, the proteins they produce interact to activate a feminizing pathway. They trigger the splicing of the feminizer FEM gene transcript, which in turn directs the splicing of the double-sex DSX gene into a female-specific form. This cascade results in the development of a female bee. However, if the queen lays an unfertilized egg, it contains only one set of chromosomes and thus only one copy of the CSD gene hemizygous. Without a second, different allele to interact with, the CSD protein remains inactive. The feminizing pathway is never triggered. The FEM gene produces a non-functional protein, and the DSX gene defaults to the male-specific splice variant. The result is a haploid male drone. Crucially, if a fertilized egg happens to receive 2 identical CSD alleles, a situation that can occur with inbreeding, the mechanism fails. The proteins cannot interact to trigger the female pathway, and the diploid egg develops into a male. These diploid drones are inviolable biological errors. The nurse bees detect a specific cannibalism pheromone released by these larvae and consume them within hours of hatching to recycle the protein. This genetic system creates a bizarre and fascinating asymmetry and relatedness within the hive. In humans, siblings share roughly half of their DNA because they receive a random half from each parent. In bees, the dynamic changes significantly because the father is haploid. When a drone mates with a queen, he passes on his entire genome to his daughters, not just a random half. Therefore, every worker bee inherits 100% of her father's genes. She also inherits 50% of her mother's genes. This means that 2 worker sisters share a significantly higher proportion of their DNA with each other than human sisters do. They are super sisters. This elevated relatedness provides the evolutionary foundation for kin selection and altruism. In evolutionary biology, an organism's primary goal is to pass on its genes. Usually, the best way to do this is to have offspring. But for a worker bee, her sisters are actually more genetically similar to her than her own potential offspring would be. The logic of evolution dictates that she can propagate more of her own genes by helping her mother raise more super sisters than she could by raising her own daughters. The hive is not a collection of individuals sacrificing for the greater good. It is a genetic fortress built to protect and propagate the super sister genome. The worker's altruism is, paradoxically, the most selfish genetic strategy available to her. To understand the mechanics of the honeybee, we must first examine the chassis upon which the entire organism is built, the exoskeleton or integument. Unlike vertebrates, which rely on an internal skeletal framework of bone, the bee relies on an external, multi-layered shell that serves 3 simultaneous functions: structural support for muscle attachment, protection from physical trauma and desiccation, and sensory interface with the environment. The integument is not a single, homogeneous material. It is a complex composite structure secreted by a single layer of epidermal cells. The outermost layer is the epicuticle. This is a microscopic, non-chitinous barrier composed of lipoproteins and waxes. Its primary physiological role is to prevent water loss. Because bees have a high surface area to volume ratio, they are prone to rapid dehydration. The epicuticle acts as a hydrophobic seal. Beneath this lies the procuticle, which is differentiated into two distinct zones, the exocuticle and the endocuticle. These layers are composed of chitin, a long-chain polysaccharide polymer, N-acetylglucosamine, similar in molecular structure to cellulose. However, raw chitin is pliable and soft. To create the rigid armor of the bee, the chitin chains are embedded in a protein matrix. Through a chemical process called sclerotization or tanning, phenolic compounds cross-link these protein chains, transforming the soft cuticle into a hard, irreversible bioplastic known as sclerotin. This material provides the tensile strength necessary to withstand the immense physical stresses of flight. The flight of the honeybee is one of the most sophisticated biomechanical feats in the animal kingdom. For decades, it was apocryphally stated that, according to the laws of aviation, a bee should not be able to fly. This misconception arose from applying fixed-wing aerodynamic calculations to an insect. Bees do not fly like aeroplanes. They fly like helicopters using unsteady aerodynamics. The bee generates lift through a mechanism known as delayed stall. As the wing sweeps through the air, it creates a massive tube of circulating air called a leading edge vortex, LEV, above the wing surface. This vortex lowers the air pressure above the wing, sucking the bee upward with a force far greater than standard laminar flow could provide. To maintain this unstable vortex without stalling, the bee must beat its wings at a staggering frequency of approximately 230 beats per second. Achieving this frequency with standard muscular contraction is biologically impossible. In most animals, muscles are synchronous, meaning one nerve impulse triggers one muscle contraction. If a bee relied on synchronous muscles, its nervous system would need to fire 230 times a second, a rate that would exceed the metabolic refractory period of the neurons. The signal would jam. To solve this, bees evolved asynchronous flight muscles, also known as fibrillar muscles. These muscles are located in the thorax, which serves as the engine room of the bee. Crucially, these muscles are indirect. They are not attached to the wings at all. Instead, they are attached to the interior walls of the thorax itself. The flight mechanism works by deforming the entire thorax box. There are two sets of opposing muscles, the dorsoventral muscles running from top to bottom, and the longitudinal muscles running front to back. When the dorsoventral muscles contract, they pull the roof of the thorax down. This flattening of the box forces the sides of the thorax outward, which levers the wing hinge up. The wings flip up. Then the longitudinal muscles contract. They squeeze the thorax from front to back, causing the roof to pop back up. This levers the wing hinge down. The wings flip down. The magic lies in the click mechanism. The thorax is constructed like a semi-rigid elastic box. It has two stable states, roof up and roof down. As the muscles pull, the thorax resists until it reaches a critical stress point, then suddenly snaps or clicks into the other state. This snapping action stretches the opposing set of muscles. In asynchronous muscle tissue, the contraction is not triggered by a nerve impulse, but by the physical sensation of being stretched. When the roof snaps down, it stretches the longitudinal muscles, which instantly react by contracting and snapping the roof back up. This creates a self-perpetuating resonance loop. The nervous system only needs to send a start signal to begin the vibration, and occasional steering signals to change the amplitude. The thorax essentially becomes a biological tuning fork, vibrating at its natural resonant frequency of 230 Hz, powered by the metabolic burning of sugar in the mitochondria. Beneath the vibrating armor of the thorax lies the internal machinery of the bee. Unlike vertebrates, which rely on a centralized cardiovascular system to pump oxygenated blood through a network of veins and arteries, the honeybee operates on an open circulatory system. The internal cavity of the bee, the hemocole, is a single fluid-filled chamber. All the internal organs, the brain, the digestive tract, the reproductive system float freely in this bath. The fluid is not blood, it is hemolymph. It is a pale yellowish liquid composed of plasma and blood cells called hemocytes. Crucially, hemolymph does not carry oxygen. In mammals, red blood cells contain hemoglobin to bind oxygen. In bees, oxygen is delivered directly to the tissues through a separate system. Hemolymph's primary function is nutrient transport, sugar, amino acids, and immune defense phagocytosis of pathogens. Circulation is driven by a single, tubular organ running along the dorsal side, the back of the bee, the dorsal vessel. This vessel is divided into two sections. The posterior part, located in the abdomen, is the heart. It consists of a series of muscular chambers with one-way intake valves called ostia. The heart pulsates rhythmically, sucking hemolymph in from the abdominal cavity and pumping it forward. The anterior part, running through the thorax to the head, is the aorta. The aorta is an open-ended tube. It dumps the hemolymph directly into the head capsule, bathing the brain and antennae in a fountain of nutrient-rich fluid. From there, the fluid flows backward through the thorax and abdomen, driven by the pressure gradient and the movements of the diaphragm, eventually returning to the heart to be pumped again. Because the blood does not carry oxygen, the bee requires a radically different respiratory architecture. It does not have lungs. Instead, it relies on a tracheal system. This is a dense network of branching tubes that deliver air directly to individual cells. The system begins on the exterior of the bee with small, valve-like openings called spiracles. There are 10 pairs of spiracles, 3 on the thorax and 7 on the abdomen. These are not passive holes. They are complex, muscular valves that can be opened or closed by the nervous system to regulate airflow and prevent water loss. Inside the spiracle, the air enters the tracheae, large tubes reinforced with spiral rings of chitin tenidia to prevent collapse. These tubes branch into smaller and smaller tubes called tracheoles, which eventually become microscopic, fluid-filled capillaries that touch the cell membranes of muscles and organs. Oxygen diffuses directly from the air in the tube into the mitochondria of the cell. To support the massive oxygen demand of flight, bees possess large expandable air sacs within the thorax and abdomen. These sacs act like bellows. As the bee pumps its abdomen in and out, a movement often mistaken for breathing heavy, it is mechanically compressing and expanding these sacs, forcing air through the tracheal system like a supercharger. Finally, we reach the digestive system, which is specialized for the processing of 2 distinct food sources: nectar, carbohydrates, and pollen protein. The mouth leads to the esophagus, which runs through the thorax and expands into a specialized organ in the abdomen, the crop or honey stomach. The crop is not a true stomach. It is a storage tank. It is a highly distensible, muscular bag capable of holding up to 70 milligrams of nectar, almost the bee's own body weight. When a forager collects nectar, she does not digest it. She stores it in the crop. The entrance to the true stomach, the ventriculus, is guarded by a complex valve called the proventriculus. This valve acts as a sophisticated filter. It has hairy, muscular lips that can snap shut to prevent nectar from entering the stomach, keeping it pure for transport back to the hive. However, if the bee needs energy for her own flight, the valve can open slightly to let a small amount of sugar pass through. Even more impressively, the proventriculus can filter pollen grains out of the nectar in the crop, bolting them into tiny pellets and passing them into the stomach for digestion while leaving the liquid nectar behind. The visual apparatus of the honeybee is a marvel of evolutionary engineering, fundamentally distinct from the camera-type eye found in vertebrates. While humans perceive the world through a single lens focusing light onto a retina, the bee perceives the world through a fragmented, mosaic lens system. This is the compound eye. Located on either side of the head, the two large compound eyes are composed of thousands of individual optical units called ommatidia. A worker bee possesses approximately 4,500 ommatidia per eye, while a drone, whose primary biological imperative is to spot a virgin queen in mid-flight, possesses up to 10,000. Each ommatidium is a self-contained photoreceptor unit. It consists of a hexagonal corneal lens on the surface, which focuses light into a crystalline cone. Below this cone lies the rhabdom, a long, transparent tube that acts as a waveguide. The rhabdom is surrounded by 8 to 9 photoreceptor cells, retinular cells, containing visual pigments. Functionally, each ommatidium captures a tiny, distinct point of light from a specific angle in the visual field, roughly 1 degree of arc. The brain then integrates these thousands of discrete points into a single coherent mosaic image. While this system offers relatively low spatial resolution compared to the human eye, visual acuity is roughly 100 times lower than ours. It provides an exceptionally wide field of view and infinite depth of field. A bee can see almost 300 degrees around its body without turning its head. The spectral sensitivity of the bee eye is shifted relative to ours. Human vision is trichromatic, based on 3 opsin proteins sensitive to red long wavelength, green medium wavelength, and blue short wavelength. Bees are also trichromatic, but their receptor peaks are shifted toward the higher energy end of the spectrum. They possess receptors for green, blue, and ultraviolet UV. This shift has profound ecological consequences. Bees are essentially blind to the color red. A red rose appears black to a bee. However, their sensitivity to ultraviolet light, 300 to 400 nanometers, unveils a hidden layer of reality in the botanical world. Many angiosperms have co-evolved to exploit this sensory channel. Flowers that appear solid yellow or white to the human eye are, under UV light, marked with distinct patterns, bullseyes, runways, and contrasting centers known as nectar guides. These patterns direct the bee specifically to the nectaries, increasing pollination efficiency. Furthermore, bees perceive a color that humans cannot imagine: bee purple. This is a mixture of UV light and yellow light, the bee's equivalent of red and blue. It is a non-spectral color, meaning it does not exist as a single wavelength in the rainbow, but is synthesized by the brain, just as purple is synthesized by the human brain. Beyond color and shape, the most sophisticated capability of the bee eye is the detection of polarized light. As sunlight passes through the Earth's atmosphere, it scatters off air molecules, Rayleigh scattering. This scattering polarizes the light, organizing the chaotic light waves so that they vibrate in a single plane. The angle of this polarization varies systematically across the sky, creating a concentric pattern of polarized bands that are always oriented 90 degrees relative to the sun. To a human, the sky looks like a uniform blue dome. To a bee, the sky is covered in a giant, invisible grid of lines pointing to the sun. This detection occurs in a specialized region of the compound eye called the dorsal rim area, DRA. In the ommatidia of the DRA, the microvilli, tiny, finger-like projections in the photoreceptor cells, are perfectly aligned in parallel rows. This alignment acts as a polarizing filter. When the plane of polarized light in the sky matches the orientation of the microvilli, the cell fires a maximum signal. By scanning the sky, the bee can determine the exact azimuth of the sun, even if the sun itself is hidden behind clouds or mountains. As long as a patch of blue sky is visible, the bee can read the polarization map and navigate with compass-like precision. Finally, we must consider the temporal resolution of bee vision, how fast they see. This is measured by the critical flicker fusion frequency, CFF. For humans, this threshold is roughly 60 Hz. If a light flickers faster than 60 times a second, we perceive it as a continuous beam, which is why movies at 24 frames per second look smooth. For a bee, the CFF is approximately 300 Hz. Their visual processing is 5 times faster than ours. This high-speed sampling rate allows them to perceive individual wing beats of other insects and to navigate through complex, moving foliage at high velocities without crashing. To a bee flying through a meadow, the world does not blur. It flows in crisp, slow-motion detail, allowing for instantaneous course corrections that would be impossible for a vertebrate brain to calculate in time. In addition to the 2 compound eyes, bees possess 3 small, simple eyes located on the vertex, top of the head, arranged in a triangle. These are the ocelli. Unlike the compound eyes, the ocelli do not form complex images. They are simple photoreceptors with massive light-gathering power. Their neural connection to the brain is extremely short and fast. The primary function of the ocelli is horizon detection and flight stabilization. Because they point upwards, they constantly measure light intensity from the sky. If the bee rolls or pitches in flight, the light hitting the ocelli changes instantly. This signal bypasses the visual processing centers and goes directly to the motor neurons controlling the wings, triggering a reflex correction before the bee even consciously realizes it is tilting. This allows the bee to maintain a level horizon automatically, purely based on the gradient of light from the sky. While vision provides the honeybee with a map of the external world, the internal reality of the hive, a dark, crowded, and enclosed cavity, is governed almost entirely by chemistry. The primary language of the superorganism is not sound or sight, but olfaction. The bee does not merely smell in the human sense. It reads a complex, syntactic chemical code that dictates social behavior, caste determination, and defense. The hardware for this chemical interface is the antenna. Far from being a simple feeler, the antenna is a sophisticated, multimodal sensory organ comprising 3 main segments: the scape, the basal segment attached to the head, the pedicel, the elbow joint, and the flagellum, the long segmented whip. In the worker bee, the flagellum is divided into 10 subsegments. In the drone, it has 11. This extra segment in the male provides additional surface area for detecting the queen's pheromones during mating flights. The surface of the flagellum is covered in thousands of microscopic sensory structures called sensilla. These come in various morphological forms, but the most critical for olfaction are the sensilla placoidea, or pore plates. These are flat, oval plates of cuticle, roughly 10 micrometers in diameter, perforated with thousands of nanoscale pores. The mechanism of smell begins when an odorant molecule, a volatile chemical floating in the air, lands on the pore plate. It diffuses through the pores and enters the fluid-filled cavity of the sensillum. Here, the bee faces a biochemical challenge. Most odorant molecules are hydrophobic. They repel water. But the sensory lymph surrounding the nerve dendrites is aqueous, water-based. To bridge this gap, the bee secretes specialized proteins called odorant binding proteins, OBPs. These proteins encapsulate the odor molecule, making it soluble, and ferry it across the lymph to the receptor site on the dendritic membrane of the sensory neuron. Once the molecule binds to the receptor, it triggers a depolarization event, an electrical spike. This signal travels down the antennal nerve to the antennal lobe of the brain. The antennal lobe is organized into spherical bundles of neurons called glomeruli. The bee has approximately 160 glomeruli. Crucially, the system is hardwired. Specific chemicals trigger specific glomeruli. When a bee smells a rose, a specific pattern of glomeruli lights up. When she smells a drop of alarm pheromone, a different, precise pattern fires. This creates a spatial map of smell within the brain. The vocabulary of the hive consists of dozens of distinct chemical signals, known as pheromones. These are classified into two categories based on their volatility and function. Releaser pheromones, which trigger immediate behavioral changes, and primer pheromones, which cause long-term physiological changes. The most dramatic releaser pheromone is the alarm pheromone. This is produced in the Koshevnikov gland, located near the sting shaft structure, and in the mandibular glands. The primary component is isopentyl acetate, IPA, a small, highly volatile ester that smells distinctly like bananas. Because it is small and volatile, it disperses rapidly through the air but also dissipates quickly. This is an evolutionary safety feature. It ensures the colony does not remain in a state of permanent hyperaggression after a threat has passed. When a guard bee stings an intruder, the tearing of the tissue releases a massive burst of IPA. This chemical lowers the sensory threshold of nearby bees, making them hypersensitive to movement and dark colors, and triggering an immediate stinging response. Conversely, the Nasonov pheromone is a signal of attraction and orientation. It is produced by the Nasonov gland, located beneath the 7th abdominal tergita plate on the back. To deploy this signal, the bee assumes a specific posture. She stands with her abdomen elevated and creates a gap between the tergites to expose the gland, while vigorously fanning her wings. This behavior disperses a cocktail of 7 terpenoids, primarily geraniol, citral, and nerolic acid. This scent acts as a homing beacon. It is used to guide lost foragers back to the hive entrance, to mark a source of water, or to signal the location of a swarm cluster to the queen. Another critical signal is the brood pheromone, or brood ester pheromone, BEP. This is a contact pheromone produced by the larvae themselves. It is a blend of 10 fatty acid esters found on the cuticle of the developing grub. The composition of this blend changes as the larva ages, effectively communicating its developmental stage and nutritional needs to the nurse bees. A larva that is hungry emits a signal that triggers the feeding behavior trophallaxis in the nurse. Furthermore, BEP acts as a primer pheromone on the workers. High levels of brood pheromone suppress worker ovarian development, reinforcing the queen's sterility signal and ensuring the workers focus on nursing rather than reproduction. The bee also utilizes a footprint pheromone secreted by the Arnhart glands located on the distinct tarsal segments of the legs. As a bee walks across the comb, she leaves a chemical trail that persists for hours, marking the comb as occupied or home. In the field, foragers use a similar chemical marker to tag flowers they have just depleted of nectar. This saves other bees the energy of landing on an empty flower, increasing the overall foraging efficiency of the colony. Finally, we must consider the cuticular hydrocarbons, CHCs. The exoskeleton of every bee is coated in a thin layer of wax that prevents desiccation. However, the specific ratio of hydrocarbons in this wax constitutes a unique chemical signature, a colony odor. This odor is derived from the queen's genetics and the specific diet of the hive. Guard bees stationed at the entrance do not recognize their sisters by sight. They recognize them by taste. They antenate incoming bees, sampling their CHCs. If the chemical profile matches the colony standard, the bee is admitted. If the profile deviates, indicating a robber bee from a foreign hive or a wasp, the guard immediately releases the alarm pheromone and attacks. The navigational capabilities of the honey bee are orchestrated by a neural architecture that defies the apparent simplicity of an insect. The bee brain is a compact organ, roughly 1 cubic millimeter in volume, and containing fewer than 1 million neurons. For comparison, a human brain contains 86 billion neurons. Yet, within this limited neural real estate, the bee performs cognitive tasks, complex learning, long-term memory formation, and vector calculus that rival those of many vertebrates. The center of higher cognition in the bee brain is a pair of mushroom-shaped structures called the mushroom bodies, or corpora pedunculata. These are analogous to the cerebral cortex in humans. They are the sites of sensory integration and associative learning. The mushroom bodies are densely packed with specialized intrinsic neurons called Kenyan cells. In a worker bee, the mushroom bodies are significantly larger relative to total brain size than they are in a drone or a queen, reflecting the worker's need to forage, navigate, and remember flower locations. The learning process in bees is associative. It is not a simple reflex. It is a conditioned response. When a forager lands on a flower and tastes nectar, the unconditioned stimulus, specific octopaminergic neurons in the brain release octopamine, a neurotransmitter functionally similar to noradrenaline. If this release coincides with a visual stimulus, like the color blue, or an olfactory stimulus, like the scent of lavender, the connection between the sensory neuron and the reward pathway is strengthened. This is long-term potentiation. The bee learns that blue equals food. This memory is stable. A bee can remember a specific flower color or scent for days, even weeks, after a single rewarding encounter. However, the most impressive cognitive feat is navigation. A forager may travel up to 10 kilometers from the hive to find food. To return home, she cannot simply retrace her steps. She must calculate a direct vector. She does this using two distinct, redundant systems, the sky compass and path integration. The primary system is the time-compensated sun compass. As we discussed, the bee uses the position of the sun, or the polarization pattern of the sky to determine direction. But the sun moves. It sweeps across the sky at a rate of roughly 15 degrees per hour. If a bee finds a flower patch in the morning when the sun is in the east and tries to return to it in the afternoon using the same angle relative to the sun, she would end up flying west instead of east. She would be lost. To solve this, the bee possesses an internal circadian clock. This biological timer is synchronized with the solar cycle. It allows the bee to continuously update her internal map. She knows what time it is, and she knows where the sun should be at that time. When she communicates a direction to her sisters, she adjusts the angle to account for the sun's movement during her flight. If you trap a bee in a dark box for 6 hours and then release her, she will fly in the correct direction relative to the current position of the sun, not the position it was in when she was captured. She has mentally calculated the sun's trajectory in the dark. The second system is path integration, or dead reckoning. This is the ability to continuously calculate one's current position relative to a starting point by integrating speed and direction over time. To measure distance, the bee does not count wing beats or measure energy expenditure, which varies with wind. Instead, she measures optic flow. This is the rate at which the visual texture of the ground moves across her retina. As she flies, the image of the grass or ground below her flows backward. Specialized motion-sensitive neurons in the optic lobes track this movement. The total amount of image motion that flows over the eye is integrated by the brain to produce a precise measurement of distance traveled. This creates a paradox. A bee flying against a headwind will fly for a longer time to cover the same distance, but her optic flow measurement will be the same. Conversely, if she flies with a tailwind, she will fly for less time, but the ground will rush by at the same total visual distance. This mechanism is remarkably accurate, but it can be tricked. If you make a bee fly through a narrow tunnel with patterned walls, the proximity of the walls increases the rate of optic flow motion parallax. The bee will think she has flown a much greater distance than she actually has. Finally, as the bee approaches the target, the hive or the flower, she switches from vector navigation, compass, and distance to landmark navigation. She uses a snapshot memory of the visual scene surrounding the goal. This is not a 3D model, it is a 2D image matching process. She aligns the current visual input with the stored snapshot. If the hive entrance was previously framed by a large rock on the left and a tree on the right, she maneuvers until the visual image on her retina matches that stored pattern. This is why moving a hive just 3 feet can be disastrous. The returning foragers will hover in the empty air where the entrance used to be, confused because their vector and snapshot no longer match the physical reality. The most famous and cognitively complex behavior of the honeybee is the waggle dance. This phenomenon, first decoded by the ethologist Carl von Frisch, represents one of the only known examples of symbolic communication in the animal kingdom outside of primates. It is a sophisticated language of vector calculus, performed in the pitch-black environment of the hive, used to communicate the precise coordinates of a resource that may be kilometers away. To understand the dance, we must first understand the stage. The dance takes place on the vertical wax combs within the hive. Because it is dark, visual cues are useless. The follower bees, the recruits, must read the dance through tactile sensation and near-field sound. They press their antennae against the dancer, detecting minute vibrations in the comb and air particle movements generated by her wings. The dance itself conveys a a direction, and a distance. The distance component. The distance to the food source is encoded in the duration of the waggle run. The dance consists of a figure-8 pattern. The bee runs forward in a straight line while vibrating her abdomen laterally. This is the waggle run. She then circles back to the starting point to repeat the run. The length of time she spends waggling is directly proportional to the distance of the flight. Roughly speaking, for Apis mellifera, 1 second of waggling corresponds to 1 kilometer of distance. However, this is not a rigid linear conversion based on geography. It is based on energy expenditure. If a bee flies into a headwind, she exerts more energy to cover the same ground. Consequently, she will perform a longer waggle run, effectively telling her sisters, "It feels like 2 kilometers, even if the map distance is only 1. This ensures that the recruits bring enough fuel honey in their crops to make the journey. The direction component. The direction is encoded through a remarkable process of transposition. Outside the hive, the bee navigates using the sun, or the polarized light of the sky, as a reference point. She knows the angle between the sun and the flower. For example, if the flower is 40 degrees to the right of the sun, that is her flight vector. Inside the dark hive, she cannot show her sisters the sun. Instead, she uses gravity as a proxy for the sun. The bee possesses specialized gravity sensors called proprioceptors located in the joints of her neck and legs. These sensors tell her exactly which way is down. In the translation of the dance, running straight up on the vertical comb represents flying directly towards the sun. Running straight down represents flying directly away from the sun. Running at an angle of 40 degrees to the right of vertical tells the recruits to fly 40 degrees to the right of the sun. The followers decode this gravitational angle and mentally transpose it back into a solar angle when they leave the hive. If the sun moves across the sky while the bee is dancing, which it does, at 15 degrees per hour, the dancer adjusts her angle on the comb in real time to account for the Earth's rotation. She is constantly updating the coordinates. There is a second, simpler dance for resources that are very close, typically within 50 to 100 meters. This is the round dance. In this performance, the bee simply runs in a circle, reversing direction periodically. This signal conveys food is nearby, but it does not provide a specific vector. It relies on the recruits flying out in random search patterns and using the scent clinging to the dancer's body to pinpoint the exact location. The communication system also includes a critical negative feedback loop, the stop signal. If a forager visits a food source and encounters a threat, such as a spider or a rival colony, she returns to the hive and seeks out other bees dancing for that same location. She produces a brief, high-pitched vibrational pulse, roughly 350 Hz, while headbutting the dancer. This signal inhibits the dancer, causing her to stop recruiting. This mechanism prevents the colony from sending more workers into a dangerous trap. Furthermore, the hive manages its labor force through the tremble dance. If a forager returns with a full load of nectar but cannot find a receiver bee, a food storage worker to offload it to, she experiences a handling time delay. If this delay is too long, she begins to tremble violently and run erratically across the comb. This dance does not recruit foragers, it recruits processors. It signals to the resting bees that the nectar intake rate has exceeded the processing rate, and more workers need to wake up and start making honey. This decentralized regulation ensures the colony's metabolism remains balanced. The construction of the honeybee nest is a feat of material science and structural engineering that rivals human architecture in its efficiency and precision. The primary building material is beeswax, a complex organic substance secreted directly from the bodies of the worker bees. Unlike wasps, which gather wood pulp to make paper, or birds that collect twigs, bees synthesize their construction material metabolically. This synthesis occurs in 4 pairs of specialized glands, the wax mirrors located on the ventral side, the belly of the worker's abdomen, specifically on sternites 4 through 7. These glands are most active in workers roughly 12 to 18 days old, coinciding with their role as house bees. To produce wax, the bees must consume vast quantities of honey. The metabolic conversion rate is steep. It requires approximately 8 grams of honey to produce 1 gram of wax. This high energetic cost drives the evolutionary pressure for extreme architectural efficiency. Every milligram of wax must be used to maximum effect. The liquid wax exudes through pores in the mirror plates and solidifies upon contact with the air, forming small, transparent scales roughly the size of a pinhead. The worker removes these scales with the stiff bristles of her hind legs and passes them forward to her mandibles. Here, the raw material is processed. She chews the wax, mixing it with saliva containing enzymes and small amounts of propolis, a resinous mixture collected from tree buds. This processing changes the physical properties of the wax, lowering its melting point and increasing its plasticity and ductility. Chemically, beeswax is a chaotic mixture of over 300 distinct compounds. The majority are long-chain esters, roughly 70%, followed by hydrocarbons, 14%, and free fatty acids, 12%. This specific chemical profile gives beeswax its unique thermoplastic properties. It is brittle at low temperatures, below 18 degrees Celsius, but becomes soft and pliable at the hive's operating temperature of 35 degrees Celsius. If the hive gets too hot, above 40 degrees, the wax loses structural integrity and can collapse, a catastrophic failure known as a meltdown. The fundamental unit of the comb is the cell. The shape of this cell, the regular hexagon, has fascinated mathematicians for millennia. The reason for this choice is geometric optimization. If you want to pack cells together on a flat plane without leaving any gaps, a process called tessellation, you only have 3 regular shapes to choose from: triangles, squares, or hexagons. Of these 3, the hexagon is the most efficient. It encloses the maximum amount of internal area for the minimum amount of wall perimeter. This is known as the honeycomb conjecture, a mathematical theorem that was only formally proven in 1999. By building hexagons, bees maximize storage space for honey and brood, while minimizing the metabolic cost of wax production. However, recent research suggests that bees do not intentionally carve hexagons. Instead, they build cylinders. When a bee constructs a cell, she builds a rough, circular tube. The bees work in a frenzy, their body heat raising the local temperature of the wax to roughly 45 degrees Celsius. At this temperature, the wax behaves like a viscous fluid. The surface tension of the cooling wax, combined with the physical pressure of bees working in adjacent cells, naturally pulls the junctions between the cylinders into perfect 120-degree angles. The hexagon is not just a blueprint. It is the physical equilibrium state of the material itself. The walls of these cells are impossibly thin, roughly 0.07 millimeters. Yet, the structure is incredibly strong. A well-built comb can support up to 30 times its own weight in honey. This strength is bolstered by the midrib, the central spine of the comb, where the cells from opposite sides meet. The cells are not placed directly back to back, they are offset. The center of a cell on one side aligns with the junction of 3 cells on the opposite side, creating a tri-pyramidal base that acts as a truss, distributing stress throughout the lattice. Furthermore, the cells are not perfectly horizontal. If they were, the unsealed nectar, which is mostly water, would run out. The bees build the cells with a precise upward tilt of 13 degrees. This utilizes gravity to keep the liquid nectar safely inside the cell while it is being cured into honey. Finally, the rim of each cell is reinforced with a thicker bead of wax, known as the coping. This acts like the rim of a paper cup, preventing the thin walls from tearing under the constant traffic of thousands of bee feet. The result is a structure that is light, strong, chemically inert, and mathematically perfect, a fortress built from the sweat of its inhabitants. The production of honey is not merely a process of gathering and concentrating plant nectar. It is a complex sequence of chemical engineering and thermodynamic manipulation designed to create a shelf-stable, high-energy fuel source that can persist indefinitely without refrigeration. To the bee, honey is winter survival. To the biochemist, honey is a supersaturated solution of monosaccharides with potent antimicrobial properties. The raw material, nectar, is a dilute aqueous solution of sugars, primarily sucrose, with varying amounts of glucose and fructose secreted by the nectaries of angiosperms. The water content of fresh nectar varies wildly, typically ranging from 60 to 80%. At this concentration, the sugar solution is unstable. It is a perfect medium for the growth of natural yeasts and bacteria, which would rapidly ferment the nectar into alcohol or spoil it entirely. The bee's objective is twofold: chemical inversion and physical dehydration. The process begins the moment the forager sucks the nectar through her proboscis into her crop or honey stomach. The crop is a specialized, distensible organ distinct from the true stomach, ventriculus. It is lined with an impermeable cuticle that prevents the absorption of sugars into the bee's hemolymph. It is a storage tank, not a digestive organ. However, the crop is not chemically inert. As the forager flies back to the hive, she secretes specific enzymes from her hypopharyngeal glands directly into the nectar. The most critical of these enzymes is invertase, also known as sucrase or alpha-glucosidase. Invertase attacks the sucrose molecule. Sucrose is a disaccharide, a complex sugar composed of 2 simpler sugars, glucose and fructose, linked together by a glycosidic bond. Invertase acts as a catalyst to hydrolyze this bond. It adds a water molecule across the linkage, splitting the sucrose into its 2-component monosaccharides, glucose and fructose. This inversion is crucial for 2 reasons. First, it increases the solubility of the sugar solution, allowing the bees to pack more energy into a smaller volume of water without the sugar crystallizing out of solution. Second, it pre-digests the fuel. By breaking the bond now, the bees ensure that the honey is immediately bioavailable for rapid metabolic uptake during the winter, when energy is needed for shivering thermogenesis. Upon returning to the hive, the forager does not deposit the nectar directly into a cell. Instead, she engages in a process of regurgitation and trophallaxis with a receiver bee, a house bee. The forager opens her mandibles and forces a drop of nectar out of her crop. The receiver bee sucks it up. This transfer happens repeatedly, forming a chain of processing bees. During this phase, the house bees perform an active evaporation behavior. They manipulate the nectar drop between their mandibles and their proboscis, repeatedly extending and retracting their tongue. This spreads the nectar into a thin film, maximizing its surface area relative to its volume. The warm, dry air of the hive rushes over this film, accelerating the evaporation of water. This mechanical bubbling can reduce the water content from 80% down to roughly 40% in just 20 minutes. Simultaneously, a second enzyme, glucose oxidase, is added to the mix. This enzyme is the preservative. It catalyzes the oxidation of a small amount of glucose into gluconic acid and hydrogen peroxide. The gluconic acid drives the pH of the honey down, typically to between 3.2 and 4.5. This acidic environment is hostile to most bacteria, including E. coli and Salmonella, which require a neutral pH to thrive. The hydrogen peroxide acts as a direct antiseptic, killing fungal spores and bacteria that might attempt to colonize the sugar solution. Interestingly, the glucose oxidase is inactive when the sugar concentration is very high in finished honey, but becomes active again if the honey is diluted with water effectively. The honey becomes a dormant disinfectant that activates only when it gets wet. Once the nectar is partially concentrated, the receiver bees deposit it into the hexagonal wax cells. But the job is not done. The water content is still too high. If the bees capped the honey now, natural osmotolerant yeasts would ferment it. The target water content is strictly below 18%. To achieve this final dehydration, the bees employ forced convection. At night, when foraging stops, the entire colony switches into processing mode. Thousands of worker bees line up in the corridors of the hive, facing the comb. They beat their wings furiously, not to fly, but to generate airflow. This creates a massive ventilation current that sucks humid air out of the cells and drives it out of the hive entrance. You can often feel this warm, moist breath of the hive if you place your hand near the entrance at night. As the water evaporates, the solution becomes supersaturated. The viscosity increases dramatically. When the sensory hairs of the house bees detect that the viscosity has reached the critical threshold, indicating roughly 17 to 18% water, they seal the cell with a fresh cap of airtight wax. The result is a substance that is chemically immortal. Because the sugar concentration is so high and the water activity is so low, the honey exerts immense osmotic pressure. Any bacterium that lands in honey is instantly desiccated. The sugar sucks the water out of the bacterial cell through its membrane, causing it to collapse and die plasmolysis. This is why edible honey has been found in Egyptian tombs after 3,000 years, perfectly preserved and safe to eat. While honey provides the colony with the raw calories necessary for flight and heating the gasoline of the engine, it is biologically insufficient for growth. A diet of pure sugar would lead to death within weeks. To build the complex proteins of the exoskeleton, the enzymes of the gut, and the tissues of the developing larvae, the colony requires a source of nitrogen, amino acids, lipids, and vitamins. This nutritional demand is met entirely by pollen. The collection of pollen is a masterpiece of applied physics, specifically electrostatics. As a worker bee flies through the air, the friction of the wind against her microscopic body hairs generates a significant static charge. A flying bee carries a positive electrostatic potential, often exceeding 400 volts. Flowers, connected to the earth, tend to hold a neutral or slightly negative charge. When the positively charged bee approaches the flower, an electric field is established between them. Before she even lands, this field exerts a force on the dry, lightweight pollen grains sitting on the anther. The pollen literally jumps across the air gap, defying gravity to cling to the bee's body. This mechanism is far more efficient than simple contact. It ensures that the bee becomes coated in pollen, even if she only brushes near the reproductive organs of the plant. Once coated, the bee engages in a complex grooming behavior mid-flight. She uses her forelegs to clean her head and antennae, and her middle legs to clean her thorax. She mixes the dry pollen with a small amount of regurgitated nectar to make it sticky. Then she transfers this sticky paste to her hind legs. Here, on the tibia of the hind leg, lies a specialized structure called the corbicula, or pollen basket. It is a smooth, concave depression surrounded by a fence of long, stiff hairs. The bee uses a joint mechanism called the pollen press, located between the tibia and the basitarsus, to compress the loose pollen into a dense, compact pellet. A fully loaded forager can carry up to 30% of her own body weight in these saddlebags. However, raw pollen is not food. It is encapsulated in an incredibly tough outer shell called the exine. This shell is composed of sporopollenin, one of the most chemically inert and durable organic polymers known to science. It is resistant to enzymes, acids, and decay. It is the reason pollen grains can survive in fossil records for millions of years. A bee's digestive system cannot easily break through the exine to access the nutrient-rich cytoplasm inside. Furthermore, raw pollen is perishable. If stored in the warm, humid hive, it would rapidly germinate or rot. To solve both problems, digestibility and preservation, the bees process the pollen into bee bread. When the forager returns to the hive, she kicks the pollen pellets off her legs into a cell, usually one located near the brood nest, not in the honey supers. House bees then take over. They ram the pollen down into the cell with their heads to remove air pockets. As they pack it, they add a cocktail of glandular secretions, regurgitated honey, and enzymes. Crucially, they inoculate the pollen with specific bacteria. These are primarily lactic acid bacteria, lactobacillus, and bifidobacterium species, which are native to the bee's crop. This mixture triggers a controlled fermentation process, very similar to the process humans use to make sauerkraut or yogurt. The bacteria consume the sugars in the added nectar and produce lactic acid. This acidification drives the pH of the pollen mixture down from a neutral 6.8 to a highly acidic 4.0. This acidic environment has three profound effects. First, it kills any pathogenic bacteria, molds, or yeasts that might spoil the food. Second, it chemically inhibits the pollen grain from germinating. The seed is killed, so it remains a food source. Third, and most importantly, the fermentation process chemically degrades the tough intine layer beneath the exine and pre-digests some of the proteins into free amino acids. The result is bee bread. It is softer, more digestible, and far more nutritious than raw pollen. The bioavailability of the vitamins, especially vitamin K and B-complex vitamins, increases significantly during fermentation. This substance is the primary food source for the nurse bees. They consume massive quantities of bee bread to fuel their hypopharyngeal glands. These glands, located in the head, synthesize the proteins from the pollen into a white, milky secretion, royal jelly. In this way, the pollen is not fed directly to the babies. It is eaten by the sisters, metabolized, and secreted as a perfect, pre-digested milk. The colony acts as a collective rumen, processing difficult raw materials into high-quality nutrition for the vulnerable young. The life cycle of the honeybee is a classic example of holometabolous metamorphosis, a complete biological transformation involving 4 distinct stages: egg, larva, pupa, and adult. This process allows the organism to occupy 2 completely different ecological niches during its lifetime. First, as a sedentary eating machine, the larva, and second, as a mobile, flying reproductive unit, the adult. The cycle begins with the egg. The queen inspects a clean, polished cell and deposits a single white egg at the base. The egg is cylindrical, slightly curved, and roughly 1.5 millimeters long. It is attached to the cell floor by a mucus secretion. Inside, the embryo undergoes rapid cleavage. Over the course of 3 days, the single nucleus divides repeatedly to form a blastoderm. On the 3rd day, the egg hatch is triggered by the dissolution of the chorion, the eggshell, by enzymes secreted by the embryo itself. Out crawls the larva. It is a white, legless grub with no eyes, no antennae, and no wings. It is essentially a digestive tube with a mouth. Its only function is to eat and grow. The growth rate is staggering. In just 5.5 days, a worker larva increases its body weight by a factor of 1,500. If a human baby grew at this rate, it would weigh as much as an elephant after 1 week. During this larval stage, the caste of the female bee is determined. This is not a genetic decision, it is a nutritional one, driven by epigenetics. For the first 3 days, all female larvae, regardless of whether they are destined to be queens or workers, are fed royal jelly. This is a protein-rich secretion from the hypopharyngeal glands of the nurse bees. It contains high levels of proteins, sugars, and lipids, as well as specific vitamins like pantothenic acid. On the 3rd day, a critical divergence occurs. If the colony needs a new queen, the nurses continue to feed the chosen larvae exclusively on royal jelly, and in massive quantities. They literally float the larva in a pool of the substance. This diet contains a specific protein called royalactin. Recent research suggests that royalactin binds to the epidermal growth factor receptor, EGFR, in the larva's fat body. This binding triggers a signaling cascade that affects DNA methylation. Specifically, it inhibits the enzyme DNA methyltransferase, DNMT3. In a worker larva, which is switched to a diet of worker jelly, a mixture of pollen and dilute honey after day 3, the DNMT3 enzyme is active. It adds methyl groups to specific cytosine bases in the DNA, effectively silencing or turning off genes related to ovarian development and queen-like characteristics. In the queen larva, the royalactin inhibits DNMT3. The DNA is not methylated. The queen genes remain on. This results in the development of large, functional ovaries with up to 180 ovarioles per ovary compared to 2 to 12 in a worker and a larger body size. The queen is not a super bee. The worker is a stunted queen. The default developmental pathway is royal. It is the deprivation of royalactin that creates a worker. Once the larva has finished growing around day 9 for a worker, the nurse bees cap the cell with a porous wax cover to allow air exchange. Inside the sealed cell, the larva spins a silk cocoon from its salivary glands and enters the pupal stage. This is the most mysterious phase. The larval body is dismantled. The tissues are broken down by enzymes into a soup of undifferentiated cells called imaginal discs. These discs are the blueprints for the adult organs. From this biological slurry, the adult structures, legs, wings, eyes, antennae are reconstructed. The compound eyes turn from white to pink to purple to black, signaling the maturation of the nervous system. Finally, on day 21 for a worker, the adult bee chews through the wax capping and emerges. She is soft, fuzzy, and pale, a tendril bee. Her cuticle has not yet hardened. She immediately seeks out food to fuel the final sclerotization of her exoskeleton. Her life as a house bee begins. For a queen, the cycle is faster, only 16 days from egg to adult. This speed is evolutionarily critical. If the old queen dies, the colony is in a race against time to raise a replacement before the population collapses. The shorter development time allows the new queen to take her mating flight and begin laying eggs as quickly as possible. The survival of the honeybee colony depends on its ability to maintain homeostasis, a stable internal environment despite extreme external fluctuations in temperature and pathogen load. To achieve this, the superorganism employs two sophisticated biological strategies: social immunity and collective thermoregulation. Unlike solitary insects, individual honeybees possess a remarkably reduced immune system. Their genome contains only 1/3 of the immune-related genes found in fruit flies or mosquitoes. This genetic reduction suggests that the colony relies less on individual cellular defense, like antibodies or phagocytosis, and more on behavioral and chemical defenses at the group level. This is the concept of social immunity. The primary mechanism of social immunity is hygienic behavior. This is a genetically heritable trait where specific workers detect diseased or parasitized brood inside sealed cells. They identify these compromised individuals through olfactory cues, specifically the smell of oleic acid and beta-ocimene released by dying larvae. Upon detection, the hygienic workers uncap the cell and physically remove the infected larva, dragging it out of the hive before the pathogen can sporulate and spread to the rest of the colony. This behavior is critical for resistance against diseases like American foul brood and the parasitic varroa mite. Furthermore, the colony constructs an external immune system using propolis. Propolis is a resinous mixture collected by foragers from the buds of trees, poplar, pine, birch. It is rich in antimicrobial compounds, particularly flavonoids and phenolic acids. The bees do not just use this as glue, they coat the entire interior surface of the nest cavity with a thin layer of propolis. This creates a propolis envelope. This envelope acts as a chemical sterilizer. It significantly reduces the baseline load of bacteria and fungi within the hive, lowering the metabolic cost of immune system activation for individual bees. When a large intruder like a mouse or a death's head hawk moth is stung to death inside the hive and is too heavy to remove, the bees encase the corpse in a thick layer of propolis. This effectively mummifies the carcass, preventing putrefaction and protecting the colony from pathogens released by decomposition. The second pillar of survival is thermoregulation. The honeybee is one of the few insects capable of decoupling its body temperature from the ambient air temperature. While individual bees are ectothermic, cold-blooded, the colony as a whole functions as a heterothermic mammal. It maintains a constant core temperature of 35 degrees Celsius in the brood nest, regardless of whether it is freezing or scorching outside. In winter, bees do not hibernate. Instead, they form a winter cluster. The mechanics of the cluster are a masterpiece of thermodynamics. The bees pack tightly together into a ball structure on the combs. This ball has two distinct layers: the mantle, outer shell, and the core, inner heat source. The bees in the mantle pack their bodies together like shingles on a roof, interlacing their thoracic hairs to create an insulating barrier. Their heads point inward, towards the warmth. These bees are effectively in a cold coma. Their metabolic rate is low, and they act purely as insulation. The bees in the core are the heaters. To generate heat, they employ shivering thermogenesis. They uncouple their flight muscles from their wings. This allows them to contract the massive thoracic muscles, the dorsoventral and longitudinal muscles, simultaneously and rapidly, without moving their wings. This isometric contraction burns honey glucose and converts the chemical energy directly into heat rather than mechanical work. The core temperature is regulated with precision. If the cluster gets too cold, the core bees shiver harder and the mantle bees squeeze tighter to reduce the surface area and heat loss. If the cluster gets too hot, the mantle expands, allowing air to circulate. The bees rotate positions. A bee on the cold outer edge will eventually push her way into the warm center to feed and reheat, while a warm bee moves out to take her turn on the front line. This rotation ensures no single bee freezes to death, provided the cluster stays in contact with honey stores. In summer, the challenge reverses. The hive must be cooled to prevent the wax combs from melting. The colony employs evaporative cooling. Foragers switch from collecting nectar to collecting water. They spread this water in thin films over the brood cells and hang droplets in the hexagonal openings. Other workers fan their wings to drive air currents over the water, absorbing latent heat through evaporation. This air conditioning system can keep the brood nest at 35 degrees, even when the external temperature exceeds 40 degrees. Finally, thermoregulation serves as a weapon. This is the heat-balling defense. In Asia, the Japanese honeybee, Apis cerana japonica, faces a predator that its European cousin cannot defeat with a sting: the giant Asian hornet, Vespa mandarinia. The hornet's exoskeleton is too thick to pierce. Instead of stinging, the bees allow the scout hornet to enter the nest. Suddenly, hundreds of workers mob the intruder, engulfing it in a tight ball of bee bodies. Inside this ball, the bees vibrate their flight muscles to generate intense heat. They also raise the carbon dioxide concentration. The temperature inside the ball rises rapidly to 47 degrees Celsius, 117 degrees Fahrenheit. The thermal limit of the hornet is 46 degrees Celsius. The thermal limit of the honeybee is roughly 48 to 50 degrees Celsius. By cooking the hornet at exactly 47 degrees, the bees kill the predator by hypothermia, roasting it alive while surviving by a margin of just 1 or 2 degrees. It is a perilous biological brinkmanship, a victory won by thermal endurance rather than venom.